Yeah, thank you, Sabri. Yeah, good morning, everyone. So I'll talk about accelerated next-generation sequencing, a step beyond traditional NGS analysis. So before going into next-generation sequencing, NGS, let's see what sequencing is. Sequencing is the process of determining the nucleotide or order of nucleotides in a DNA fragment. Next-generation sequencing, or NGS, help us massively parallelize this process and generate large amount of data in a short time. Unprecedented growth has led to the generation of exabytes of next generation data annually, and it is expected to hit zettabyte scale this year. So this large amount of data brought us new opportunities and challenges. Opportunities, the decreasing cost made NGS a viable option in routine clinical practice, meaning we can sequence more and more patients and use them in the clinical diagnosis process. Also, availability of large-scale population datasets enable us to implement powerful machine learning applications and gain new insight at population level. However, there's a challenge. The traditional NGS tools show long processing hours, and this poses a limitation. For example, long turnaround times limit the use of NGS in time-sensitive clinical applications. Also, processing delays can restrict the scalability of population-level NGS analysis. So let's talk about the hardware used to run NGS analysis. So the traditional best practices tools use CPUs for computational power. And due to the inherent nature of these CPUs, it takes hours and sometimes days to complete a single analysis, single sample. So this is a challenge, and scientists and engineers have addressed this by developing accelerated hardware platforms. So Illumina, or DRAGEN from Illumina, and Parabricks from NVIDIA, optimized for GPU, are two such accelerated platforms. What is the difference between CPUs and these hardware accelerators? CPUs allow us to run hundreds or 150 or 200 processes in parallel, but the hardware accelerators allow us to run thousands or tens of thousands of processes in parallel. That is the competitive edge we get with the accelerated hardware platforms. DRAGEN use field programmable gate arrays to optimize for NGS analysis. These are specialized chips that can be programmed to run a specific process. DRAGEN allow us to use FPGA to run NGS analysis. Parabricks is from NVIDIA. It is an application layer that help us run GPU, NGS on GPUs. Since this is a application layer, Parabricks is the application layer, we can use it to run an analysis on different GPU platforms. For example, L4, A100, H100 are three different GPU platforms with different complexities and performances. Parabricks allow us to run NGS on all these different GPU platforms. So scientists have evaluated these hardware accelerators for NGS analysis, and have demonstrated that these can be used to reduce the processing time. But they have not done a side-by-side comparison. And also, importantly, they have not looked into the computational resource usage of these NGS and accelerated NGS analysis tools. So that is what we started to tackle. We developed three pipelines: CPU-only pipeline, which is the best practices, industry standard best practices pipeline we used as the baseline, and DRAGEN and Parabricks are the accelerated pipelines. So we developed these two, Parabricks and DRAGEN, following the best practices as well as the guidelines provided by the vendors. So here we focused on key performance matrices, which we will talk later in the presentation, and we evaluated the scalability of accelerated pipelines. Also, we identified potential bottlenecks and looked for strategies for further improvements and again further higher speed ups. So one of the interesting part in the accelerated compared to CPU-based pipeline is to compare the runtime. So we used whole genome data to compare these different pipelines. And in this slide, you can see the hours, number of hours used to run our test samples. So you can see the key take-home message in this slide is that as expected, CPU shows the longest runtime, but with any kind of accelerated platform, the runtime dropped drastically. So we can see that with the accelerated platforms, the runtime can be cut down effectively. So this can be quantified. This performance gain can be quantified by calculating speed up. It helps us to quantify the performance improvements gained by the accelerator hardware. We can calculate it. Here we divide the CPU only runtime by the accelerated runtime. So what does this say? This says how many times faster a process will run when we implement it on an accelerated platform. So let's compare the speed up of DRAGEN and Parabricks in different stages of a NGS workflow. In an NGS workflow, in a basic NGS workflow, there are two main stages. We call them mapping and variant calling. And in this slide, we are comparing the speed ups gained in different accelerated platforms for these two stages. As you can see, in mapping, as the performance of the GPU increases, the speed up also increased. For example, L4 to H100, the speed up increased from 14 to 30 in mapping. Those are the highest speed up observed for the test data set. And DRAGEN and Parabricks on H100 mapping showed similar speed ups. But the other interesting feature we observed here is compared to mapping, variant calling stage, one of the last stages of the pipeline showed comparatively higher speedups. For example, on H100 with Parabricks for mapping, the maximum speedup we observed was 30, so 30-fold speedup. But on the same GPUs with the same data set, variant calling reported 126 speed up. So that is a big difference. And another interesting feature, the DRAGEN showed lower speed ups in variant calling compared to Parabricks with different GPU platforms. Then we studied the scalability of accelerated platforms. Here we studied how speed up could vary with the coverage. So coverage is, you can think of it as the size of the data set, higher coverage, larger data set, lower coverage, smaller data set. So, and higher coverage of a higher resolution than the lower coverage samples. So if we compare, when we study the speed up and variation, how speed up varied with this coverage, we could see that during the mapping on DRAGEN and H100 Parabricks, the speed up increased with the coverage. We saw that the mapping on DRAGEN and H100 scaled up with the coverage. However, the variant calling step showed the opposite pattern. The speed up decreased with the coverage. So variant calling was not scaling up with the size of the dataset or coverage of the sample. Then it is the interesting part where we studied how these accelerated platforms, especially the Parabricks, use computational resources available in the system. Here we used one of the tool developed by Norwegian AI cloud engineers. This tool called Job Analyzer allows us to interrogate a process running in a system and look at resource usage at given time points. So we studied how the GPU usage varies across different time points in the Parabricks runs. And we observed at a given time, only a small fraction of the GPUs available in the system is utilized by the Parabricks. Also, we tested on multiple samples and studied the maximum GPU usage per sample. That was also a smaller fraction of the available GPUs. That means significant proportion of the GPUs are not utilized during the Parabricks runs. what does that ask? That raised the question, was that this small fraction of the GPUs used? Was that the optimum GPUs required to run this analysis? Or can we improve, optimize this tool to further utilize more GPUs and gain higher speedups? We also studied the disk usage during the Parabricks run. So what we saw was during the mapping process the Parabricks use higher disk read-write rate. In other words, Parabricks frequently read and write from the disk compared to the variant calling. And we aligned this disk usage with GPU usage time series data, and we observed that when the Parabricks is reading, writing frequently from the disk, the GPU usage drops almost down to 0. So there are frequent drops of the GPU usage in the Parabricks, in the mapping process. So what does that mean? When the Parabricks start reading and writing from the disk, the GPU goes into idle time. And remember, we compared the speed up of mapping and variant calling. Mapping speed up was much lower than the variant calling speed up. That is why. So we observed that the idle times in the mapping process is the reason for the low speed up we observed. Then we so in the previous slide, we talked that variant calling was not scaling up with the coverage. So we thought, okay, let's see how the GPU usage varies with the high and low coverage samples, or large and small data sets. So as the data set, the coverage increases, or larger data sets, the GPU usage was lower for all accelerated callers. So that means the callers are not utilizing the GPU. So it is the same question. Was that the optimum amount of GPU required to run the variant calling, or was that, or can we improve further? So that's what we tried with our optimized accelerated variant calling workflow. What we did here was to split the whole genome into different sections and run them in parallel. So what we saw was interesting, what was expected as well. When with the optimized workflow, we saw a higher GPU usage, and the speed up was also comparatively higher than the original. So this was the cartoonish depiction. The numbers are also quite impressive. So you can see the mapping, variant calling with original Parabricks workflow used GPU 8 to 15% of the available GPUs, and reported speed up of 75 to 127. But when we compare the speed up of the optimized workflow, GPU usage was much higher. It was around 18 to 42% of the GPUs were utilized during the Parabricks run, and most importantly, the speed up jumped to 245 to 329. This is a comparable jump from what we observed in the original. So we demonstrate that we can further improve the speed up by optimizing these workflows. Then we thought of a practical framework to select an accelerated platform. So first we did a concordance analysis. What we did here was to study the validity of the accelerated variant callers. So here we compared the variant calling from the accelerated workflows to the variant calling from CPU-only best practices standard pipelines. And the concordance, the recall and precision work much higher for both Parabricks and DRAGEN. What does that mean? That means we could essentially replace the variant caller from a standard CPU-only best practices with an accelerated workflow and reach the similar results, which was what would the researchers and experts would expect. So the validity of the accelerated callers are quite high. Then we can compare the cost of running accelerated callers. We can compare. So we ran DRAGEN on a local server at Oslo University Hospital, and A100 Parabricks on Fox, Dagfinn introduced a little while earlier. They were local infrastructures. And the H100 and L4 were set up on Norwegian AI Cloud with Google Cloud Platform as a backend. So these were on the cloud infrastructure. So we can compare the cost of this local infrastructure and cloud infrastructure. And this is what we observed. The DRAGEN cost was quite high, much higher than the Parabricks on A100 on the on the local infrastructure, but the performance were similar. And in the Norwegian AI Cloud, the Google Cloud Platform, H100 were expensive, much more expensive than L4. Also, they offered much higher performance. So we can talk about pros and cons. So there is a text-heavy slide, but the take-home message is over there. So what we saw, DRAGEN is highly specialized for Illumina sequence analysis. So DRAGEN is offered by Illumina, so they are tightly coupled. And why do we say this is specialized to run and highly specialized to run sequence analysis? Over here, you can see in the DRAGEN column, there are many check marks, meaning that they offer at least one tool or one module for each and individual step in the workflow. But here in your number 5, X in the DRAGEN, meaning that it won't work with other sequencing providers. It will work with Illumina. Parabricks is versatile. It offers greater options, but lacks several key tools. So what we see over there in several rows, you see 2 check marks, meaning they offer multiple tools, so multiple modules. So we can either choose the module or tool that works our use case, or we can use both and get the intersection to reach higher precision. And also, importantly, the number 5, different technologies, a check mark means it will work not only with short-read Illumina data, but with long-read other sequence providers, PacBio and NanoPo. And there are a few X's in the Parabricks column, meaning they lack tools for those steps, but in the current version of Parabricks, but we can hope that they will release those tools in the future releases. And I only presented a high-level overview. So this study was accepted for publication in Bioinformatics Advances. This is not online yet. So follow our Nike and Noura LinkedIn pages. We will share the link as soon as it is online. So finally, the future directions. We are looking forward to provide this as a service for industry and academia and SMEs. Also, we are looking forward to further optimize the accelerator tools and release open source bioinformatic workflows, providing greater flexibility for our users. And we are looking forward to implement accelerator tools on Nike infrastructure with large data sets so that comprehensive machine learning studies can be performed. I would like to thank Norwegian AI Cloud, Norwegian National Unit for Newborn Screening Center, and Argonne Leadership Computing Facility in US. So we performed a reproducibility study with US, so I didn't talk about it today. And the Scientific Computing Service, Division for Research, my current workplace. And we have our co-authors of the study over there. Thank you. Oh, that's a question.
Thank you very much. Just a short question regarding the platforms used. When will we get a European-based platform and not using US-based platforms for the NGS sequencing?
So this A100 will run on Fox. Yeah. So and Nike.
And this will be also for the rest of the European countries? Because I assume they will have the same challenge, or at least that they are compatible with each other so that you can compare, for example, and harmonize across the European Union for the sequencing data.
So this A100 is on Fox, and access to Fox for other European is I'm not the right person to answer. So.
Transfer your workflow to Lumi, for example, for European.
Yeah, okay. So the
But
Lumi.
any collaboration with the other hospitals doing the same NGS sequencing, setting up the same?
So Norwegian, the Newborn Screening Center, Oslo University Hospital, they were running DRAGEN. So we were collaborating with them. And so Ivan Hovig, he has a setup in Australian Radium Hospital. So we have been collaborating with them. So this is more of a collaboration in the Norwegian setting. And the reproducibility study we did with US, we wanted to see how the Parabricks works in one system compared to another similar system, but in two different settings. Yeah. Thank you.